Hello my geniuses, welcome to another biochemical lecture series. Today it's a great science tutor Lorenzo. I'll be talking about the HMP Shunt, or we call it the Hexose Monophosphate Shunt, or the Pentose Phosphate Pathway. It has a lot of name. It has the phosphogluconate pathway too, different name that people gave it. So this pathway is very important because the goal is that we are forming ribose sugar, pentose phosphate pathway. We are producing five sugar molecules, which is ribose, ribulose, xylose, every sugar with five molecules. That's what we want to produce at the end of this video. I want you to stick to the end because it's very important. I'll be giving you useful mnemonics you use for your exams, and when you write the exam you pass it solely. So if you are just new to my channel, make sure to subscribe and turn on your post notification button so that anytime I drop any video you'll be sure to be notified. And if you want me to make a video for you, drop it down in the comment section. When I look at it, I'll make that video for you. So let us jump right into the video. So the first thing we'll be talking about now is what is HMP Shunt? Why is it important? What is there? What makes it so useful to us? Because now we know that when glucose is absorbed by the tissue, which is the muscle, what does it do? It undergoes what we call glycolysis. So anytime glucose is absorbed through the help of insulin, we call it what? Glycolysis. So it undergoes the glycolysis by being phosphorylated. It becomes phosphorylated to glucose 6-phosphate, fructose 6-phosphate, down to forming pyruvate. And if oxygen continues to be fed into the cell from pyruvate, it enters into the mitochondria and continues for TCA cycle, entering into the ETC, which is electron transport chain, for making what? ATP. But for here now, you are not talking about glycolysis pathway, but we need the glycolysis pathway because there's a bypass here. Are you getting me? There's a bypass here, and oxygen is not directly needed here. Oxygen is not directly needed. We have oxidation. In this pathway, it's an alternative form to oxidize glucose. Naturally, we are oxidizing glucose. Oxygen is fed into glycolysis to the help of what we call oxidative phosphorylation. So oxygen is used to drive the glycolysis pathway. Are you getting me? Oxygen is used, but in this pathway we are also oxidizing glucose, but we are not using oxygen. We are using what? NADP plus. Don't forget. So we are using NADP plus. We are not using what? NAD plus. We are not using oxygen. We are just using NADP plus. So this is an alternative pathway. Alternative pathway in which glucose can be oxidized. Glucose can be oxidized, so there will be bypass of fructose 6-phosphate. Are you getting it now? So number one, it's an alternative pathway for oxidation of glucose. An alternative pathway for oxidation of what? Glucose. So we are oxidizing glucose not with oxygen but using what? NADP plus. So that is the first thing you have to know. Very important. I told you that another name for HMP Shunt is phosphate PPP, which is pentose phosphate pathway. Don't forget, pentose phosphate pathway, or we call it Womberg-Dickens pathway. Womberg-Dickens pathway, or we have phosphogluconate pathway. Anyone, they have different names. Are you getting now? So the key product to HMP Shunt is what? NADPH. Are you getting now? The raw materials which you use for oxidation is NADP plus, but the product at the end of this reaction is to make what? Pentose sugar. Is to make what? Pentose sugar, or we call, which called the ribose sugar or the pentose sugar, and NADPH. That's the goal, because ribose sugar is important for formation of RNA and DNA. Are you getting now? And NADPH is very important because it provides reducing power for biosynthetic reaction and what? Protection against oxidative stress, because NADPH is important. The body sometimes has a reduced oxygen, the body is always faced with oxidative stress. So that is why NADPH comes to help us to shield by reducing the oxidized molecule in the body. So when the oxidative stress is coming, the body will reduce it instead of continue to be in its oxidized form, because if it continues to be in oxidized form, it will form what we call radicals, and radicals will damage the body, increasing inflammatory response in the body, and we don't want that. And the reason for the ribose sugar, the ribose 5-phosphate, which we'll be getting, which is the ribose phosphate, is used for nucleotide, nucleic acid, and coenzymes. You get it now? So very simple. That is the goal. That is the goal, and the end product of what we are talking about today. Number one, it's an alternative pathway for oxidation of glucose. We do not need oxygen, but you need what? NADP plus. That is one thing you need to know. Are you getting that? And the end product here is to get NADPH and what? Ribose 5-phosphate. NADPH and what? Ribose 5-phosphate. Ribophosphate is used in nucleotide, nucleic acid, and coenzymes. The second question asks ourselves, where is the site? Where is it produced from? Now, it all occurs in the cytoplasm, but cytoplasm of different places. Can be cytoplasm of the liver, any place where HMP shunt is needed, that is where it is produced to. So in the liver, adipose tissue, adrenal cortex, testes, and RBC. So it's very, very important in RBC, very, very important, because if NADPH is not produced in RBC, the RBC will undergo hemolysis. It will be scattered, it will be destroyed, and it will cause different problem. So NADPH is important in RBC because it prevents the action of oxidative stress or oxygen radicals on the RBC, causing hemolytic anemia. So it's very, very important. The liver, the adipose tissue, the adrenal cortex, the testes, and the RBC. So why do we need HMP shunt? That's the question you should ask yourself. Why do we need HMP shunt? Now, our RBC do not have mitochondria. That is one thing you should know. Our RBC do not have what? Mitochondria. So since it doesn't have mitochondria, it depends on HMP shunt for what? NADPH production. So it doesn't have mitochondria, so it depends on HMP shunt for NADPH production. Are you getting that? So if there's production of NADPH, what will happen? It will maintain what? The reduced glutathione. So glutathione, we don't want glutathione to be in its oxidized state. We want glutathione to be in its reduced state. So that is why, since there's absence of mitochondria, for us to maintain glutathione in its reduced state, we have to produce NADPH. If not, glutathione will be in its oxidized state, and if it's in oxidized state, it will cause oxidative damage of the red blood cell, causing what? Hemolysis. That is why RBC needs it very well. Now, another question to ask yourself is that what is the stimulating factor? What stimulates HMP shunt formation? One, well-fed state. When you eat, glucose is absorbed by the tissues, and when glucose is absorbed by the tissues, it can form HMP shunt. And when you eat, immediately the body releases insulin. Insulin helps you to break down the food to produce ATP and to form other products too. So immediately also tyrosine is also released. Tyrosine is very, very important because it increases basal metabolic rate, and here 100% we need oxygen. We don't really need oxygen directly, but we need oxidation using what? NADP plus. So what is the inhibiting factor here? When you are fasting, starving, or you're having a strenuous exercise, your body lacks oxygen, your body cannot oxidize anything, your body do not have glucose, so HMP shunt will not occur. Also, glucagon is released when you are stressed, but in this particular point you are well fed, and well fed state causes increase in what? HMP shunt. And the last one, cortisol, also is a stress hormone that is released anytime there is what? Stress, and which favors which inhibits HMP shunt, rather, which inhibits HMP shunt. But this one is welfare state that favors HMP shunt. So now, what is the function of HMP shunt? If we can ask ourselves, why is there HMP shunt? Why is it important for the body? Number one, I told you, formation of nucleotides. Formation of what? Nucleotides. I told you that the ribose sugar here, which is ribophosphate, is important in what? Nucleic acid formation, DNA and RNA synthesis. Shebi? DNA and RNA synthesis, because you have nucleotides, and in nucleotides you have what? Nitrogenous base, which is purine and pyrimidine. You have phosphate, and you have the sugar, which is your ribose and ribulose, or ribose and deoxyribose. Are you getting now? So you are in your nucleotides, you have your nitrogenous base, you have your phosphate group, and you have your ribose sugar. I get him now. And the ribose sugar is either ribose sugar or you are using deoxyribose, depending when you want to form DNA or RNA. So the next one is formation of NADPH. We know we need NADPH here. Why? Because it helps to keep oxidized substance in their reduced form. We don't want them, because when they are in the oxidized form, they become bad. You get it now? So they help to protect RBC by maintaining glutathione in their reduced state. We talked about this before. If not, they'll cause hemolysis. They also use towards synthesis of steroids. Also, when we are producing cholesterol too, we use NADPH as part of the substrates too. Fatty acid too, when we are producing fatty acid synthesis, but when we are doing the malic enzyme, when citrate shuttle leave the mitochondria, we need NADPH. So throughout the process of fatty acid synthesis, we need NADPH throughout. Also, other lipids and other steroid hormone, we need NADPH. Also, we need it for detoxification of xenobiotics, because NADPH is useful in the cytochrome P450 system, because cytochrome P450 system help in detoxification in the liver. Again, it helps in detoxification, like in drug metabolism, detoxification of toxic substance, inactivating foreign substance. Because if foreign substance enter and it gets absorbed into the blood, it goes to the liver, and what the liver does, it secretes so many cytochrome P450 enzyme, or so many cytochrome enzymes that detoxify the chemical substance, preventing them from having harms in the body. Okay, now, so like I said, in short, HMP Shunt provides what? NADPH, which helps in reducing power, and ribofosphate, which helps in nucleotide synthesis. Are you getting now? So very, very important. Now let us go to what we call the biochemical pathway. So hope you are following me. The biochemical pathway. So the biochemical pathway is divided into two parts. Are you getting now? You have the oxidative phase and the non-oxidative phase. The oxidative phase and the non-oxidative phase. The oxidative phase is called the irreversible phase, which means you cannot go back, you are only going one way. The irreversible phase. And first of all, we'll start, and it occurs in the cytoplasm, like I talked about it. And the goal of the oxidative phase is to produce NADPH and also to produce ribose 5-phosphate. NADPH and ribose 5-phosphate. You are looking at it on your screen. It's so important. Now, first of all, let us start with the step 1. Are you getting now? So let's say, at first of all, what happened? Glucose is phosphorylated at carbon 6. Should we? Glucose is phosphorylated at carbon 6 to produce glucose 6-phosphate. Glucose is phosphorylated at carbon 6 to produce glucose 6-phosphate. Now we have glucose 6-phosphate. Instead of glucose 6-phosphate to go to fructose 6-phosphate by phosphohexoisomerase, instead of that, it bypass. It doesn't go there. Are you getting me now? So anytime you eat, some part of the glucose gets stored as glycogen through glycogenesis. Some part of the glucose continues to form pyruvate through the help of what they call glycolysis, while some glucose gets shunted by bypassing fructose 6-phosphate to go and form what? NADPH and what? Ribose 5-phosphate. You get it now? So that is it now. So remember, the glucose 6-phosphate will now form, instead of forming fructose, it doesn't form fructose. It now goes and gets what? Oxidized. So it becomes oxidized and form what we call 6-phosphogluconeolactone. Are you getting now? 6-phosphogluconeolactone. That's what it will form. So oxidation of glucose 6-phosphate forms 6-phosphogluconeolactone, and now which we reduce NADP plus to NADPH. Again, 6-phosphogluconolactone is formed when glucose 6-phosphate is oxidized. So when you oxidize glucose 6-phosphate, you get 6-phosphogluconeolactone by the enzyme glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase, whereby reducing NADP plus to NADPH. Now the second one here is that now you have 6-phosphogluconolactone. It is now hydrolyzed. 6-phosphogluconolactone is now hydrolyzed to 6-phosphogluconate. 6-phosphogluconate. So 6-phosphogluconolactone is oxidized to 6-phosphogluconate by the enzyme 6-phosphogluconase. 6-phosphogluconase. You get it now? 6-phosphogluconolactone is hydrolyzed to 6-phosphogluconate by the enzyme 6-phosphogluconase. Nase is ending it because of there's hydrolysis there. Now since you have 6-phosphogluconate now, 6-phosphogluconate, now what happens? 6-phosphogluconate is now oxidized to ribulose 5-phosphate. Don't forget, 6-phosphogluconate is now oxidized to ribulose 5-phosphate. So we started as glucose 6-phosphate become oxidized to 6-phosphogluconolactone. Then we hydrolyze 6-phosphogluconolactone to form 6-phosphogluconate, hydrolysis. Then now we oxidize 6-phosphogluconate to form ribulose 5-phosphate. Now when we are using a dehydrogenase enzyme. Why? Because we oxidized, we didn't hydrolyze. So when we oxidize, what happened? We will oxidize 6-phosphogluconate to ribulose 5-phosphate by using the enzyme called what? 6-phosphogluconate dehydrogenase. And remember, anytime we have a dehydrogenase, what happens? It's an oxidoreductive enzyme. So anytime we have it, we are reducing NADP plus to NADPH. So we have gotten NADPH in two. The first one, when glucose 6-phosphate oxidized to 6-phosphogluconolactone, we get NADH there. Now when 6-phosphogluconate is oxidized to ribulose 5-phosphate, we get another one because NADP plus is reduced to NADPH. Now ribulose 5-phosphate, to end the oxidative phase, now is cleaved by two enzymes. One is cleaved by, it is isomerized. First of all, if you isomerize ribulose 5-phosphate, ribulose 5, it is a ketose sugar. So if you isomerize ribulose 5-phosphate, you get ribose 5-phosphate. Isomerization of ribulose 5-phosphate give us what? Ribose 5-phosphate. Why epimerization of ribulose 5-phosphate give us what? Xylulose 5-phosphate. So we have isomerization of ribulose give us ribose, why epimerization of ribulose give us what? Xylulose. Are you getting now? Are you getting now? So now the key point to note here, it is this phase, this oxidative phase is irreversible. That is one. And there's a generation of 2 NADPH. I told you 2 NADPH in the first reaction when glucose 6-phosphate is what? Oxidized to 6-phosphogluconolactone, and when 6-phosphogluconate is oxidized to ribulose, you get 2 NADPH. Are you getting now? Then you also release what? 1 CO2. So 1 CO2 is also released at the end of this reaction. 1 CO2 is always released at the end of this reaction, and the end product is what? Ribulose 5-phosphate. The end product is what? Ribulose 5-phosphate. Are you getting now? So when you are using glucose 6-phosphate, you are getting what? Ribulose 5-phosphate, 2 NADPH, and you are giving out CO2. Are you getting now? So that is what we have talked about. So remember, we have 2 NADPH releases CO2, produces ribulose 5-phosphate, and all these things are important for what? Biosynthesis, antioxidant defects. Are you getting that? Defending the cell from antioxidant action, nucleotide synthesis, and the what? Detoxification. Now the next step, which is the non-oxidative phase. The next step is the non-oxidative phase. And in the non-oxidative phase, what is happening here? We call this a reversible phase because you can go back to the beginning and you can go back to the end. Anyone is a reversible phase. And the goal of this is to produce what? Sugar, or to produce intermediate of glycolysis. That is the goal of all these things here. So the first step here is 1. What are we doing now? We have ribose. I get it. We have ribulose 5-phosphate is isomerized to ribose 5-phosphate, which we talk about, and if I epimerize ribulose 5, I get what? Xylulose 5. So we have talked about that. So the first part of this non-oxidative phase is what? I get it now. We have what's called transketolase reaction. What is transketolase reaction? Very, very important. Now we have ribose 5-phosphate and ribulose 5-phosphate. See it on your screen. Ribose 5-phosphate and ribulose 5-phosphate. If you use the enzyme transketolase, what will happen? If you use the enzyme transketolase, which means you transfer what we call 2 carbon sugar molecule for transketolase. When you are using transketolase, this is a transketolation reaction. When you are using transketolase in the presence of thiamine pyrophosphate, which is vitamin B1. So when you are using thiamine pyrophosphate as the coenzyme, are you getting now? And you are using a transketolase enzyme, what are you doing now? You are transferring 2 sugar molecule, 2 carbon sugar molecule from the keto sugar, which is xylulose 5-phosphate, to the aldose sugar, which is ribose 5-phosphate, which means you are taking 2 sugar molecule from what? Xylulose, and give it to ribose. When you give it to ribose, it will form what? Siduloheptose. I get it, man. Siduloheptulose, which is 7 carbon sugar molecule. You form 7 carbon sugar molecule because when 2 is given to 5, it becomes what? 7. So sidoheptulose 7-phosphate, which is a ketose sugar, and glyceraldehyde 3-phosphate, which is an aldose sugar. So 2 carbon molecule is transferred from xylulose 5-phosphate to ribose 5-phosphate to form a ketose sugar, which is sabdoheptulose 7-phosphate and glyceraldehyde 3-phosphate. That's a transketolation reaction, which is 2 carbon molecules transferred in transketolation reaction in the help of TPP, which is thiamine pyrophosphate. Now the next one is transidolation reaction. Aldolase, transidolation reaction. What happens in transidolation reaction? In transidolation reaction, using transaldolase, you are transferring what we call 3 carbon sugar molecule. Now 3, not 2 carbon sugar molecule. 2 carbon sugar molecule is for transketolation reaction, 3 carbon sugar molecule is for transidolation reaction. So we are transferring 3 carbon sugar molecule from sadoheptulose. Now are you getting that? Always from the keto sugar, not from the aldose sugar. You always remove it from the keto sugar. The first one is we are taking 2 from xylulose to ribose 5-phosphate. If we transfer 3 from heptulose 7-phosphate to what glyceraldehyde 3-phosphate, it will give us fructose 6-phosphate, which is a keto sugar, and erythrose 4-phosphate, which is an aldose sugar. That's adolation reaction using thiamine pyrophosphate as a coenzyme. The last one we are doing is also transketolation reaction. So we have transketolation reaction, transadolation reaction, transketolation reaction. In this transketolation reaction, what is happening now? We are now combining fructose 6-phosphate plus what? Erythrose 4-phosphate. Now are you getting now? Plus what? Erythrose 4-phosphate. So if we combine it again, we still get the what? Sadoheptulose 7-phosphate and glycerol 3-phosphate. Are you getting now? Sadoheptulose 7-phosphate and glycerol 3-phosphate. Don't forget. So this reaction is so important here, very important, because the goal of this reaction is that we will be getting what we call, we'll be getting fructose 6-phosphate. Are you getting now? Glyceraldehyde 3-phosphate. This is what we are getting, and once we are getting glyceraldehyde 3-phosphate, we are donating it into the glycolysis cycle. We are donating it into glycolysis cycle. Do you get it now? When we get glyceraldehyde 3-phosphate, it will become an intermediate of what? To become an intermediate of what? Glycolysis. Are you getting that? It will become an intermediate of what? Glycolysis. Don't forget, become an intermediate of glycolysis. So when we break down sadoheptulose 3-phosphate, you give 3 to what? Glyceraldehyde 3-phosphate. It will form fructose 6-phosphate and erythrose 4-phosphate. Are you getting that? And so when we do this now, what happens? We are forming what? Glyceraldehydes. Are you getting now? Glyceraldehyde. So glyceraldehyde is an intermediate of what? Glycolysis. Are you getting now? So one thing you have to remember, that this is a reversible phase, and we are not forming any DPH and CO2. CO2 is not being produced, any DPH is not being produced. Are you getting now in this one? And it involves transketolation reaction and transadolation reaction. And here there's what? An interconvertible between the pentose sugar to the hexose sugar. So you are forming fructose 6-phosphate at the end of this reaction, you are forming fructose 6-phosphate at the end of this reaction, and you are forming glyceraldehyde 3-phosphate at the end of this reaction, which are intermediate of glycolysis. Are you getting me? So this is very simple. So the help of HMP Shunt 1 is to produce NADPH, give off CO2, NADPH, give us CO2, and also produce ribose 5-phosphate, or we call it ribulose 5-phosphate, that get isomerized to ribose 5-phosphate. This is what the end product. We got ribulose 5-phosphate. It is a keto pentose sugar. It's a keto pentose sugar, while ribose phosphate is an aldopentose sugar. So that's the end. And also we are getting sugars that are intermediate of glycolysis. You get it now? So that is the goal of all this reaction. You get it now? So when we get ribose 5-phosphate, we use it as one of the components in nucleotide, nucleic acid, and coenzyme formation. And anytime we get NADPH, we use it to protect the cells from what? Antioxidant action. Use it to carry out so many biochemical reactions, like we said in the functions. That is the goal. So I hope it all made sense to you, and you have understood everything I've talked about here in the HMP Shunt. So keep watching my video, like and subscribe to my YouTube channel, and share it to every medical student that needs this video. You get that? Don't forget that. Help me share it out to everybody. You get that? I need the channel to move forward so that I can push this video more to people that need it, because I like making medical videos for medical students, because when I was there I see most people struggling. So now I just said that you make videos for them so that they will understand in the simplest way possible. See you in my next video. Bye bye.